Okay, so welcome back to this next video in which we are discussing acetylcholinesterase enzymes and also acetylcholinesterase inhibitors. Okay, so we're now on the acetylcholinesterase inhibitors. So we've discussed the non-covalent inhibitors. So we've discussed edrophonium, tacrine, and donezapyl, which all bind ionically to the glutamine at position 334 within the anionic site of the active site of the acetylcholinesterase enzyme. This results in the acetylcholine not being able to come into the enzyme and therefore a decrease in the function of the enzyme. However, this is an ionic interaction and therefore it's not a particularly strong interaction, so the drug will continue to come in and out of the active site of the enzyme, so it will continue to flip between a state of inhibiting the enzyme and not inhibiting the enzyme. So the enzymes also will flip between being inhibited and not being inhibited. So the enzymes will still be degrading acetylcholine whilst they're in this not inhibited state, and therefore these drugs do not completely stop all degradation of acetylcholine. Instead, what they do is they slow down hugely the degradation of acetylcholine. And we've seen how edrophonium is used to diagnose myasthenia gravis. So what you do is you take a patient who you suspect has myasthenia gravis and you'd give them a small dose of edrophonium, and if they responded well to it, i.e. if their muscle weakness, their ability to contract their muscles went up, then that would indicate that that would be a positive result, and it would indicate they do have myasthenia gravis. Because if the edrophonium helps, then that indicates that what is happening is that you still have functional nicotinic acetylcholine receptors on the sarcolemma. It's just that you have far fewer of them, and therefore you need a bigger acetylcholine signal to actually produce the same level of activation of the muscle cell in response to axon stimulation. Okay, so then tacrine and donezapyl are used in Alzheimer's disease, at least in the early stages of Alzheimer's disease, to increase cholinergic neurotransmission within the brain and increase cognition and memory. Okay, we're now going to move on to the covalent acetylcholinesterase inhibitors, and we'll start with those that are reversible. So the three drugs in this category that I'd like you to know the names of are neostigmine, pyridostigmine, and physostigmine. Neostigmine, pyridostigmine, and physostigmine. And these drugs all are going to put something known as a carbamoyl group onto the serine at position 203. So let's see what these drugs are going to do. Okay, so basically these are carbamoylating agents. So let me turn over the page and show you what these are going to do. So firstly, let me tell you what a carbamoyl group is. So I'll write down the name of it first. Carbamoyl. Okay, so these are based on a structure known as carbamic acid. So I'll show you the structure of carbamic acid first, and then I'll tell you what a carbamoyl group is. So carbamic acid has a carboxylic acid group here, okay, and then it has an amino group of that carboxylic acid group, like so. So this is the structure of a carbamic acid molecule. Okay, now carbamoyl groups are based on the structure of carbamic acid, but they're slightly more general. So a carbamoyl group can have arbitrary R groups in these positions of the hydrogen. So you can have any old group there and any old group there. So these drugs, neostigmine, pyridostigmine, and physostigmine, they all have these carbamoyl groups, like so, where they have this carboxylic acid with a nitrogen linked off it, and then these two arbitrary groups off the nitrogen. Now, what's going to happen is these drugs are going to interact with that serine at position 203. So let me show this again. Here is this alcohol group on serine at position 203 in the esteratic site of the active site of our acetylcholinesterase enzyme. Now, what these carbamylating agents are going to do is they're going to react with this serine hydroxyl group at position 203, and they're going to form an ester link between this carboxylic acid group here and this alcohol group. So what's going to happen is the alcohol group is going to come fully off the carbamoyl group, the hydrogen is going to come off the alcohol of the serine, and then you're going to link that carbon to the oxygen of the serine 203. So what you're going to end up with is the serine 203 enzyme, now with this carbamic acid or carbamoyl group, because it's not actually a carbamic acid. It could be have any old groups in this R and R prime position. Okay, but the point is that the serine hydroxyl group now has this carbamoyl group on it. We say that the serine has been carbamoylated. Okay, so this is a carbamoyl group. Now, basically, once this carbamoyl group has bound to the serine at position 203, it's not easy to break it off again. It's really, really difficult to hydrolyze this ester link between a carbamoyl group and an alcohol group, basically. So this bond is not easily hydrolyzed. So you remember that when we were discussing the function of the acetylcholinesterase enzyme, we discussed how at the end stage, if I get the picture up to remind you, where is it? Here. At the end stage, you end up with this acetyl group bound to the alcohol group of serine at position 203, and then water comes in and hydrolyzes this bond to regenerate the enzyme again. Okay? When you have a carbamoyl group linked to the serine at position 203, this group is not easily hydrolyzed, so it will remain bound to the serine at position 203 for very long periods of time, far longer than the acetate would. So, the question is, if you have this group stuck on the end of your alcohol group of the serine at position 203, is the enzyme going to be able to function? Well, the answer is no. Because remember, the serine alcohol group was essential in this nucleophilic attack on the acetic acid group of the acetylcholine molecule. It attacked this carbon here. And with the carbamoyl group attached, it will not do that, basically. So the enzyme is not going to function. So these drugs, neostigmine, pyridostigmine, physostigmine, which all come and carbamoylate that alcohol group of the serine at position 203, they're all going to make this covalent modification, for it's a covalent bond that links this carbamoyl group to that alcohol group. And this is going to inactivate those enzymes which have undergone this covalent interaction. Okay, so these three drugs then, neostigmine, pyridostigmine, and physostigmine. Neostigmine and pyridostigmine are both used as treatments of myasthenia gravis. So what are they going to do? They're going to be given orally, okay, and they will go into the synapses at the neuromuscular junctions, okay, and they will inhibit those acetylcholinesterase enzymes in the neuromuscular junction that, remember, were in these groups of 12 and were attached to the heparan sulfate proteoglycan within the basement membrane of the neuromuscular junction. Okay, and then when the alpha motor neuron releases acetylcholine into the neuromuscular junction cleft, then the size of the amount of acetylcholine that will actually make it to the sarcolemma is firstly going to be much more, because it won't be broken down nearly as much by the acetylcholinesterases in the cleft. And secondly, that acetylcholine will remain in the cleft for a much longer period of time. So basically, you hugely amplify the acetylcholine signal, and that now is great enough to produce stimulation of these fewer nicotinic acetylcholine receptors that are on the sarcolemma. Okay, and that at least goes some way to treating the muscle weakness that we see in myasthenia gravis. Okay, the final drug, physostigmine, is not used to treat myasthenia gravis. Instead, it's used to treat glaucoma, and I will explain to you what glaucoma is in the next video. Okay.